Dr. Sunave, our program director, to kindly address the audience.
Hello, good morning, all of you. Uh, this is the first ICE workshop we have planned at Chile. Welcome all of you. And we have with us today uh, distinguished guest, Sri Sanjay Wandekar sir. He is the center head Chile College. And he wanted him to be uh, guest to launch this first workshop on this because I think he's known as his last TV. I heard that Dr. Saraswat was talking about him as a Rudra man. Okay. So that whatever current supercomputers are there in India, a couple of kind of computing power. I think it's all because of him. Whatever Paramrudra systems he has built, and within a few months, actually, we'll be having the topmost machine in India, which is the 20 petaton. And it's all making India efforts, whatever his vision and direction he has given. I think that has made impact on uh, India's supercomputing vision. So he's a real visionary and execution person. So and sir, just want to tell you that ICE is also indigenously developed software, and that's why we want you to be launching this workshop. And we are also going to contribute and take way with not just ICE but all other HPC applications. So thank you, sir, for coming here, and I would like to welcome him with him. Just to give you background about the ICE workshop, I think under MSM also we have this mandate of proliferating technologies, softwares, tools developed under NSM and various other projects. We are going to make it more popular, creating a user base for that. I think that is very essential for all of us. So we are trying to build a community for life sciences, which can uh, get benefited with these activities. And because
is it audible now?
yeah,
Thank you. So I think I'll just start again. So I'd like to congratulate our bioinformatics team for this workshop, which is on ICE. And I think just now, Dr. Uddavesh talked about the ICE features and other things. And throughout the day, you will be having more details on the ICE platform and the applications, bioinformatics applications, which are available on this platform. So initially, I'll talk about the national supercomputing mission. What is that we have achieved till now, and what is that going to be there in this mission? So till now, we have already deployed about 40 petaFLOPS of compute power across the country in various academic and educational institutions, apart from CDAC. And the largest machine which is going to come up is about 20 petaFLOPS, which is going to be there as a national facility and will be accessible to all the users across the country. So total, we are actually envisaging about 90 petaFLOPS of compute power to be established during another maximum 9 to 10 months timeline. So, so this is part of our national security mission infrastructure side. So apart from building the infrastructure, we are building the complete HPC ecosystem within the country, starting with the development of various technologies which are required for building the supercomputer, which includes our Rudra servers, network, software stack, and so on. So that is what is required for building Paramputra, which is already established in few of the systems here across the country. So this is one part of the ecosystem which we have developed, which also includes the manufacturing of these systems within the country. So that is another thing which we have done. The next thing which under ecosystem is we are developing various indigenous applications in various other areas, including urban environment, urban modeling, where we are actually uh, giving support to the cities in terms of uh, what is the weather. Means weather forecast is one part of it. Flash floods within the cities, then pollution control. All these things are available in as a one central ecosystem where the urban people can see this as well as uh, the authorities can see and take actions on that. This is one application which is developed indigenously. Second one is basically flood forecast, where we are actually working with various rivers, basins, and there we are again giving flood forecast. Third one is oil and gas exploration. Tools for required for this is another thing which we have developed under this mission. Then fourth one is basically tools and applications for materials and chemistry research. And the one which we are now interested is basically bioinformatics tools and softwares. So under this bioinformatics tools and softwares, we have developed drug discovery tools like Tango, TangoDoc, Depict, Cumulate, and so on. Under genomics pipeline, tools like NGS, Vivida, these are the tools which are being developed, and these are available for the user community. So now it's available to the user community in a very easy manner. We have developed this Ice Cloud, which uh, is going to increase the user base because users may not know how to use the HP systems. So here, this Ice Cloud is a very user-friendly environment. People can start using this HPC systems very easily with this uh, front end and Ice Cloud. So I am very happy to see this workshop being conducted here today to proliferate all these bioinformatics tools along with the Ice Cloud, which is going to be available for the users to use this. So again, I would like to congratulate bioinformatics team for their efforts on this development of various tools and Ice Cloud, as well as now proliferating all these efforts to the user community. Thank you very much.
Thank you, sir. And just want to thank you again. And we are committed if with your leadership and maybe DG sir, what he was planning earlier with the Rudra servers and then Bharat OS and then maybe creating a national cloud. We are not sufficient. Maybe multi-center approach is required, and your vision and DG sir's vision will be required if we really want to have something as a national cloud. So we can just complement there or contribute there in this in future direction. Thank you. Thank you for coming.
Thank you. Thank you. So please continue. I think with this workshop.
Yes. Thank you for launching.
Thank you.
Deep Malviya, so I request him to kindly start with his presentation. Hello, good morning, everybody. We are going to talk about the integrated computing environment for life science, that is ICE Cloud. So the first question comes is, what is ICE and what is ICE Cloud? So ICE Cloud is a platform which is built indigenously to provide cloud services for research and development. And the whole idea is that using this platform, the research become reproducible. Because when the researchers does something, the same environment we can produce or simulate in the ICE so that other user can seamlessly check and produce the same result in the same environment. This cloud is built with focus on efficiency, security, and scalability. It integrates multiple key components that streamline data management, computing, collaboration, and workflow automation. So one of the key principles behind the development of ICE Cloud is that we have to provide ease of access to computational resources. Am I audible? Okay. So why ICE? There is lot of demand from the research community. One of the demand is we need to provide dynamic provisioning of compute and storage. Many researchers, especially PhD scholars, they have laptop at their home in lab, but sometimes they need more high computing power that they only need in their labs they available, and lab resources are also limited. So using these ICE, they can run a tool which requires high memory, high CPU on their demand at their browser by getting access to ICE Cloud. We have provided dedicated container repository for user applications. We have a secure data repository within the territory of the country, because the key idea is that the researchers' data should lie in the boundary of the integral territory of the country. So the data which you are uploading to the ICE or sharing with each other is guaranteed that it is within the boundary of the India. And lot of researchers publish many tools. Those tools are different languages like C, C++, Python, Java. So what happens if they want to share that tool? So that particular version of Python or GCC compiler or JDK version has to be installed on their system. So at least 1 or 2 hours initially always invested of other people to run that particular tool to at least for the first time. And this, if you will, that tool is used by 100 users, so you count the number of research hours invested just to run the tool. By suppose 2 hours one user taking 100 users, it will be 100 into 2 hours of research. The very good researchers investing in only this running the tool. So this is really waste of the research mind in the country. So with ICE, we are solving this particular problem. Also, lot of researchers need their website to be hosted on for the public access. Researchers want to share their data to the people, but they don't have a very secure or a good platform where they can do all those things very seamlessly. So we provide on ICE Cloud, research can provide their public hosting of their website or application or tool on the platform. Also, the tool or the containerized tool which they will run, that also supported with the dynamic computing environment. Like they can manage the number of CPU, number of memory, how much CPU or RAM they need to provide for to run that particular containerized application. So the strategic advantages of ICE, as already mentioned, this ICE is built indigenously at TDAC Pune. It provides indigenous excellence. It has technological synergy. ICE provides based on your compute load, it can scale dynamically based on the load. So your application today you need 4 GB of RAM, 2 CPU. Tomorrow you can double it. Day after tomorrow you can triple it. Then data sovereignty. As I said, data lies within the territorial boundary of the India, and it has lot of economic impact, because every researcher, every student cannot purchase high-end laptop. Using ICE Cloud, even the simple minimum laptop configuration, they have seamless access to highest computing power as a supercomputer in the country. So we offered many services like ICE Compute, ICE Storage, ICE Collaboration. Because suppose I have big amount of data on my system, I want to share with other people. So first, another researcher has to download and upload in his tool, then he has to run. So with collaboration, he can just share the data by controlling the access and to which particular user he is going to share and for how much time. So the owner of the data has complete control of his sharing access. It's not that once he shared, he lost the control. He can revoke this access any moment of time. User can push or upload his build tool in a containerized form at the ICE Cloud, and other user can seamlessly use it. And not this, but the people or researchers in the country can directly develop their code on the ICE platform. They can run their code. They can build container image there itself on the ICE platform, push it to ICE Cloud and host also. Everything is uh, directly available at the ICE Cloud. Additionally, for every stages, whatever the compute power will be required, that is available through this cloud platform. So as I said, we have iStorage where you can share, collaborate. Here, large file also you can upload easily. So the with advanced upload mechanism like S3 object storage tool, user can upload large file. ICE computer, I have already mentioned, it is dynamic environment. You can specify the number of CPU memory when you will run it. And you can run the third party repository, like some tools are available on GitHub repository, some is on Docker repository, some tool are on Quay repository. So any tool which is publicly available, you can run directly on the ICE platform. It's not necessary that tool has to be lying on the ICE platform. So it is a compatible for other image repository in the world. As I said, I already discussed that with iShare. What iShare, you can see it's like your private Google Drive. Only thing is, even in Google Drive, once you share it, you while sharing it in the iCloud, you can mention for how many time I have to share my data. Suppose you want to share for 3 days, then you can share. After 3 days, it will be automatically revoked from the user's access. So that is one advantage. Additionally, I provide, because as a research community, it is very important that the owner or the absolute owner of the user should have his complete control of his access mechanism. Also, once he share, instead of 3 days, he want to make now 2 days, he can change those things anytime. As I said, we have a dedicated container repository where any tool a researchers or any user build, he can upload to ICR like ICE container repository and run on the platform. So I will just start a little demo of ICE Cloud. The screen is visible. So this is the home page of the ICE Cloud. So on right side, you have login button. So I'll just log in. So this is the once user login, this is the first dashboard he see. As you see, there are various different services uh, available to ICE users, and it is every service is a role-based access. Once user is given role, he can access only those services. So as I told, it is your private drive. Here there are 3 drives: my drive, shared drive, and public drive. My drive is like my private data. Only I have absolute control of my private repository. The shared drive means if some else user has shared any data with me, I can access it under shared drive tab. The third is public drive. As a host of the ICE Cloud, the CDAC can share his any data to other users in a public drive. So that data is available to all users. When they log in, everybody will get access to that data. Only thing is, here you can only read this data. You cannot write to public directory in the storage ICE public drive mechanism. And here you can see there is a different options. But when user want to upload anything, user can select any file or folder and he can upload it. The only thing is you can upload not just any particular file, but also the folder. And when you will be uploading data, the thing is in research community or in now in almost the data is associated with metadata. So when you will be uploading data, you can associate metadata with it. Like if I say any file, which and I say so you can add as many metadata fields. Later on also you can edit those metadata fields. This will help you to search your document later on. So data is uploaded and it is available directly here. So this is the data which I had uploaded. You have download functionality, so all normal Google Drive functionality is there. Here there is a for ICE, we have one more important thing is like quota. Whenever user will log in, you will see you will have one quota. So this quota is like you can request quota to the admin of the ICE Cloud, and you can request that I want this much CPU memory or RAM. Once request has been submitted, we can approve the request, and you will have that much CPU quota. As the resource is bound, so you will have those particular limited access of that quota. But on a particular case, different user can be given different types of quota. So this is one important thing that the community should know. There are different terms and conditions. The data hosting people are hosting data here. One thing is guaranteed: that data is sovereign. You are the absolute owner of your data, and CDAC. The data is lying within the territory of the CDAC country. I mean, and you have user profile. You can see your quota also, your profile. And I said every service is associated with role. So user has particular role, only those service he can access. So suppose you want to run HPC application, you will have role. You want to storage, you will have role. For compute, you will have role. And uh, as I said, that we have ICE container repository. So these are the tools which are directly available at ICE Cloud, which you can use and run it. Simply, like my colleague Ramakrishnan will give you a complete hands-on to how to use the ICE compute. So right now, I explained the basic nutshell of the ICE Cloud. The additional how to use the compute tool, Ramakrishnan will uh, going to explain you. Later on, you can ask any question which you have. So Ramakrishnan will join you.
Yes. Okay.
Good morning to all. Now I'm going to do the demo of the ICE, and I'm requesting to everyone to everyone log in their user credentials, and if you have any problem, chat with us. Our technical team will support. So, okay. Right now I'm
Your user accounts are created, right? So we log in. Hope everybody has their login credentials. So kindly log in onto on the main page. Okay. Yeah. So now if you
Okay.
see your screen, this is how we can log in.
okay. Now I'm going to log in. When you open the home page, there is a login button is here. So when you
mention home page, the ICE Cloud got it.
yes, our okay.
So in your browser, open your browser page and type this, as you can see on the screen.
You can type the icecloud.in. It's a website name. When you enter, and you can see that right side, top right, top right, you can see the login button. So click the login button, and you,
Hope everybody is on the login page.
okay.
Okay.
Now I'm logging with my username. So I log in with my user account.
Hope After registering on Ice Cloud, everybody must have received the login.
That already.
It's already done. So hopefully everybody is on the dashboard now. Yeah.
Oh, okay. Once you got the dashboard, you can see that we have the storage and containers. Two important menus are there. And the storage, Sandeep will already talk about that, how to upload the files and everything. So I'm briefly talk about the storage. So we have the My Drive, it's our personal drive, and this is shared drive. Some files anybody shared with me, those files will be displayed here. And third is a public drive. It's like a common data sets we are displayed here for this workshop. So you can get all the details in the public drive. And the public drive is read-only mode. Now, I'm talking most about the container. In the containers, we can create a lot of containers. And if you go to the container and the image section, we have a lot of images are curated images are listed here. Curated list of images here, like a basic Ubuntu available, or any Jupyter-based data science notebooks are available. And
Just wait.
Yeah, 1 second.
okay.
Kindly check your email IDs after registering. You might have got the login credentials. And this is just an introduction to what all are the features.
Yes.
However, after this talk, everybody will be again restarting with the login page itself. If, in case you have missed it, just
This is right
wait
now
for
just
the-
introduction only. Actual hands-on will come after this talk.
So before starting the actual hands-on, we'll have a check on everybody's account is functional or not. So please now let us go ahead.
Okay. Now here the curated list of images are we have, and it's not only this list. We can bring it from the Docker Hub. Any images you want to run in the ICE, we can do that also. So right now, for a simple example, I will be choosing the Ubuntu is the first image. So I click the launch button. Once it's launched, you can see that there is how much memory and the CPU allocated for this Ubuntu image. So I give one CPU and 1 GB RAM. So click that create container. Now the container will be created. You can see that this page will show all the your existing containers. In my account, there's a lot of containers already running. Okay. Now refresh the container list. You can see that our Ubuntu is now up and running. Now right side there is 3 dots are there. Click that one and use the connect. Select the connect option. Once the connect option selected, you can see that now we have the terminal of Ubuntu. And this Ubuntu, lot of tools are pre-installed, like a screen command and all other tools.
Does anybody have any questions? The online audience? Everybody present here in the lab, they are in sync with whatever is going on? Yes, ma'am.
So,
Okay.
somebody got created.
Ask people, everybody is able to log in and see at this dashboard. Show them the dashboard.
Those who have already registered for Ice Cloud login, if they have not received the emails, they'll receive it shortly. the confirmation emails. So currently, if you are not able to do the things what are being currently displayed on the screen, we are having the hands-on session which are there. This is mainly showing the features. Once the hands-on session starts, we'll go in lines once you are also able to do the things. Right now, just
Okay.
get a look and feel of, get used to the look and feel of icecloud.in.
Okay, now I'm started the Ubuntu container. When you see the starting the Ubuntu container, here you can see that your data is mounted in the slash drive RAM key, and your public data is are connected slash drive public, and that locations are your all the files are available. What you see in the storage area, those files are is mounted in this location. So when you see here, drive. Yes, it's like now your normal Ubuntu terminal. You can run all your Ubuntu commands, or you can run here. And if any commands are not there, you can use the package manager like an app. You can install those tools. So when you use the ls command, you can see the all the files present in this folder. And you can see that my there is a 3 folders are present: RAM key, public, and shared. It's exactly same of 3 container for 3 folder for your private data, public data, and shared data are available. And I'll go back back to our cloud homepage. Here I can click the image section. You can see a lot of other images also there. So another example, I'll show you the one data science Jupyter notebook. Suppose you want to develop any Python based ML AI applications or anything you want to do Python. Jupyter notebook is one of the prerequisites mostly people are using. So in this case, here Jupyter data science notebooks are available. Here most of the tools like a scikit-learn or matplotlib, lot of pandas, all tools are pre-installed. So launch that data science notebook, and again you
We can see messages on the activation of accounts. So slowly all the accounts will get activated. So now we are having just a walkthrough of how to launch a Jupyter notebook. Anyways, when we'll be actually doing some exercise, this would be repeated.
yes. So now I'm click the create container. Now in the behind the scenes, the Jupyter container will be created and it will be connected. All your data sets also again same way out Ubuntu terminal is connected. Similarly, the Jupyter notebook, all your data are connected. Now here in the Jupyter notebook row, you can see the password. Copy the password that may needed for next step. Then connect the Jupyter notebook. On the Jupyter notebook connected, they are asking the token. So paste the token which you copied it earlier. Then click the login. Once you connect that, now our Jupyter notebook is running, and you can see the website, our private data, shared data, and public data are mounted. And once click the Python kernel, we can run normal any commands. You can execute like a normal, how you can use a normal Jupyter notebook, and this Jupyter notebook will be created. So first, it's mandatory that go to your folder, then create the your Jupyter notebook, because that data will be persisted in this folder. So when I say, I'll give the name is a test. Now on my Jupyter notebooks are saved. If we come to our cloud dashboard, under the storage section, we can see the file drive. Here that file will be created. This is a file. That Jupyter notebook file is created here. So the data in the Jupyter notebook will be persisted even after our container will be deleted. Suppose if you delete the container, that still the data is will be persisted. And apart from that, in our ICE platform, we have GUI containers. Suppose you have some legacy tools, similar like a VMD. The VMD tool won't work on the browser. So we build pre-packaged with the container in the GUI tools also. So I'm launching the VMD now. And we in similar way, we can view how much memory we want to assign for that particular container. So I'm giving 2 CPU and 3 GB RAM. You click the refresh button for the get the status of the container right now. So now my VMD containers are running. You can see that. So copy the password, and as usual, you connect the container. GUI container. So you can see that a VMD is available in the desktop. So when you double click, you can open the VMD. Like a VMD, we are pre-packaged a lot of containers like IGV and a lot of other tools. So we can use those tools. We can access our data. Here also, our data are mounted. We go for the drive and RAM key, public chat. Again, those 3 folders are mounted. So every container you are using, same data sets are across all the containers are mounted, and you can work on parallel also. So this is one of the features of ICE. And once your workloads are completed, suppose my VMD visualization, my part is completed, you delete the container. And every user, there is a quota is assigned. If you try to use more than that, it will be show the error. So in this case, you delete the old containers and start the creating new container. So I'm deleting my VMD container. Similar way, I can delete my Ubuntu container also. So if you multi-select the containers, you can see that here top side we given the one button. We delete the multiple containers once. Yes. We need to make sure that your data will be stored on flash drive and you are using it. If you store outside, it won't be persistent. So make sure that your data will be stored on those your personal drive. Private drive.
When you load the instance, it's for that instance there is a path that will go away once you close the pod, what we saw. So whatever operations you do, every time make sure whatever your outputs are, what you require later on, must be saved in /drive/username. And that is what you see here when you click on file type. That is what you see. So everything there will remain even if the pod is closed. Yes, it's completely. You get this.
Okay. Now this is a overall overview is done.
This was just to show everyone that GUI enabled tools as well as command line tools, for everything there has been something provisioned in terms of these pods which can be launched and we can do those operations.
And for those who want to know about the Linux command line, we prepare the one file. So go to your public drive. Here there is a command.pdf. Here we'll give the step-by-step how to execute the Linux commands. So you can see the overview and you can follow this step. You can understand the entire command line. Okay, thank you.
Okay, so now please the users who are online, kindly raise your hand to let us know whether you are able to log in into ICE Cloud. Okay, Rutuja, what is your query? Please unmute Rutuja. Okay, but from where can I change? Okay, yes. Okay, Khushi, are you able to log in? Okay, I'm just calling you. I'm just calling you some names. Okay, Khushi, are you able to log in? Okay, we just has everybody received has their login credentials? Okay, I request I'll call out the names. Just unmute yourself and tell us whether you have logged in, you are successfully logged in or not. Okay. And we have posted the registration process video. Hope you all have followed that while registering. I request only those who are not able to log in up till now to message that login pending. Just message that on the chat.
इसका पेंडिंग
Nilesh sir. Nilesh sir. Nilesh sir, we are just yes sir. Yes. Have you
Yeah, I've not received.
Nilesh sir?
I had registered, but I have not received a login credential.
अभी अपन आगे जा सकते हैं अगर किसी को कुछ लॉगिन कर चुका हो
Nilesh Karkar sir. Right.
Okay, we'll proceed with the next item back.
Just walking through this, that whoever has not yet registered at all on iCloud, once you type icloud.in, you get this page, and once you click on login, there is this create new account who have not got any email messages from ICE. The first step to with a register account is to be done first, then you will be given the login credentials. Just in case there are no more email communications by iCloud any user. Okay, sir. Nilesh sir, we haven't any registration from you. Kindly just the email
Not
ID was
able to hear you.
sometimes.
I didn't hear you properly. Please.
Hello.
Hello.
Am I audible now?
Yeah. So do I need to re-register it?
So we'll wait for another 3-4 minutes and then we'll start with our next session.
Okay, okay, okay. Fine.
where we'll be having first small introduction to all the users on one of the first applications, that is NGS. It would be a presentation, and followed by that we'll have hands-on on the NGS analysis. So I request you all to kindly let us know in the next 4 minutes if you have any. I have any.
Okay, okay, okay.
Okay, so I'll just repeat myself. Am I audible now? So in the next 3-4 minutes, kindly let us know if anybody has any issues. We'll be having the next talk on the NGS applications that can be done, and Sucheta would be presenting an overview of that. Followed the overview, we'll be having a hands-on session on the same. So we'll be starting in next 5 minutes. Mr. Manish, kindly type your issue in the chat box.
No, ma'am, I don't have any issue. I
Okay,
have successfully logged in.
People without any issues, then those who have raised hands, then they can unraise them. Yeah, Dr. Anisha, you have any
Dr.
query?
Anisha.
Okay.
Okay, so we'll be now starting with the next talk by Dr. Sucheta Patil. So I'll ask her to kindly take over.
Yes, so good morning, everyone. I hope everybody is able to see my screen, and I hope I'm clearly audible to everyone.
Yes.
Yes. So let's get an intro. Hello? Hello?
It's not on. Akash? No, here people can't hear. नहीं, कुछ बात ही नहीं करो.
We were having some issues with the speaker mic here. Anyways, I'm continuing my presentation. NGS data analysis on the cloud. Okay, I'm sorry, we are just waiting for a few more minutes.
And one of them, it's alone.
Hello?
This is a loose connection of the PC. On है?
हाँ तो ऑन है किसी को ऑन है हेलो
Sorry. You just talk to them like what you are going to discuss. Just
I hope everybody has been
चल रहा है
Yes, so the next session is going to be about the next generation data analysis.
Yes, Sucheta, you may start. Yes. Yes, sir. You start.
So, good morning, everyone. Now we are going to start with the analysis of NG using Ice Cloud. So, let us have a brief introduction. Let us have a brief introduction to cloud and how NGS data can be analyzed on it. So we will be looking at next generation data, how it is analyzed, unique features of Ice Cloud over other online services, and some popular NGS analysis pipelines. More specifically, we'll be looking into steps of PCF analysis, that is variant calling. Please. And how Ice Cloud is different from desktop. Really very sorry for the technical glitch. Yes, so we were looking at how next generation sequencing technology is helping research in life sciences. So basically, whatever DNA sequence analysis is being performed, it mostly helps revolutionize research requiring agricultural advancement, crop breeding, etc. A lot of it is being used in medicines, where a lot of clinical research is being carried out for drug discovery, identification of new biomarkers, and a lot of work is going into environmental sciences. Yes, some people are having problem hearing my voice. Am I audible to everybody? Everybody is able to hear me clearly. So we saw what were the implications of NGS and how important it is for our day-to-day research activities, which are influencing our daily life from agriculture to the environmental sciences. However, it requires a lot of expertise for bioinformatics analysis, and these tools and databases, understanding of these tools and databases is required. Uh, most of you are obviously very much familiar with these databases and bioinformatics tools, but even then, there are certain challenges which come for newly evolving researchers or scientists in this field, where you have to set up your own server for analysis of bioinformatics data. Since we all know that most of these tools are Linux based, a lot of high investment is required for setting up Linux servers, right? And there can be limited access to these tools if you're going, if you're working outside your institute, if you're traveling, and all of a sudden you want to share your data with somebody. It is a lot of hurdle there, where you have to be very much on your desk all the time to analyze your data, right? And another challenge is sharing of your data, since most of these raw files that are generated by the sequencers are in gigabytes, terabytes of data, and transferring that data to another institute for sharing requires downloading the data on your own hard disk and then transporting that hard disk to another institute, again uploading the data to their server, and then you start the analysis. It takes a lot of efforts, time, patience into transferring your data. Another thing is not just the data transfer, you also require a lot of knowledge about how to install these software on your server, which requires a lot of software compilation knowledge, a lot of IT skills are required into installing these tools. And lastly, another challenge, the biggest challenge is downloading large bioinformatics databases and storing them on your server, which requires again a lot of fast internet. Sometimes some databases take weeks, months to download. And again, every time you want to analyze your data on some different server,
you again have to start with download this. So all these things require a lot of patience and takes a lot of skills, highly IT skills to get started with your bioinformatics. Once you get started, it's not a big deal, but before starting your analysis, you have to put a lot of effort to set up your system, to set up your databases, etc. And again, sharing is again always a problem. So now, so I'm getting a feedback that my voice is lost. Please. Yeah, sorry for the pause. Yeah. Thank you everyone for being patient with this presentation. So we saw what all challenges we have in NGS data analysis using your own offline servers. Now let us see how iCloud can help you with your analysis. Firstly, it is easy access for all the new users. It's a very easy access for bioinformatics. It has a very simple and user-friendly interface compared to other online servers like Galaxy. It is a single platform for secure storage and analysis. Most of the online servers do not provide you storage for a long time, or it's temporary storage, and it's not very fast, fastly accessible. Also, since these online tools are hosted on other servers in other abroad countries. So this is our Indian server where you have the same platform to store your data as well as analyze your data. Then it also supports both genomics and structural biology. So this is another plus point of using Rice Cloud. Most of the other online services, they are dedicated to genomics or structural biology. You can't do both the analysis on one platform. Then next, it is also user friendly for both basic users as well as advanced users. So basic users can reach the bioinformatics tools with the click of Docker images, and advanced bioinformatics applications where you require to use the Visual Studio Code, etc. for code development. You can use ICE Cloud. Next, multiple ways of file upload to web browser are available. So you don't have to use any command line for uploading your data, or you don't have to zip your files or zip your folders for uploading. You have got multiple ways of uploading, and that too, it is a high-speed upload. Next, simple interface for compute requisition. So if you know if your server or desktop has got a limited compute availability, that is RAM and storage that is present on your server is quite limited sometimes, and you need to expand your analysis. So you have to start from purchase of hardware. Whereas on iCloud, you can simply request for more space in the for storage as well as RAM memory. Then another plus point is sharing of your files with the registered users. So it's a very highly secured storage that we are providing you, and you can securely share with another users, like specifically for clinical data, which is sensitive, has information in it. So you can share it with specific registered users only. And another lastly benefit is there can be some data you want to publicly. It's not a sensitive data, but you want to share it with the world. As Sir has talked about it, you can post your own tools. You can develop your own tools, deposit it on iTunes, you can post it on your even with registered users. So this is about how we can get benefit from iCloud. So starting with the NGS data analysis pipeline. So firstly, I will talk about this variant column pipeline. So it is the most popular pipeline available in genomics, used in genomics. So it has multiple steps. We will be going through this pipeline. and we'll be having a hands-on tutorial also on this. So I'm just giving you a brief overview. So it starts with quality tuning of data, and followed by reference index, followed by read mapping to the reference genome. Then we sort the map and perform variant calling in those files. And finally, we look at the functional annotation. So next, we'll be coming up with a case study also. So let's see how this looks, how the files look. So first step is said you have fast queue trimming, that is your raw reads trimmed. So this graph basically shows you the quality of your fast queue reads, how they are before trimming, that is low quality bases are present in the raw reads, and after we trim it, we get a good and clean data. Now this clean data summarizes as having best quality for per base sequence, etc. And all the tools that we're using are trim galore for this tutorial. Then next step is reference genome alignment. Since we are looking at the reference based alignment for VCF analysis, we have a reference genome. Reads will be aligned, and this has multiple steps in it. So firstly, we have to index the genome using a tool like a roadkill aligner or HiStat tool. That indexed genome is then used for mapping the FASTQ files, that is the clean raw reads. And these clean raw reads, when mapped onto the indexed genome, will generate a SAM file. Then this SAM file is then converted to BAM file using SAM tools, and followed by that, also index it for visualization of CF files in future. So the next step, we'll have a look at SAM file. You have a header region and a bottom region, and it tells you how each and every read is aligning to your reference. And particularly this column is quite interesting. It shows you it is the cigar string that is there, which shows how the read is mapping. So for example, here you can see these are the reference positions for your read. So this is your read, these are the reference positions 1 to 19, and its representation is here. After aligning, it is having one insertion over here. It is having 3 matches, one insertion, again 3 matches, one deletion, and again 5 matches. It can be represented as 3M, 1I, 3M, 1D, 5. So this is how multiple files that are generated in this file are to be analyzed or interpreted. So this was one example for it. Next, you convert the SAM output to BAM files, where you require files followed by indexing. And after indexing, you can use integrative mix viewer to actually view these files in the browser. So we are providing you activate the active IGV tool. You can open your own and upload these files and results. And yes, the variant analysis also on these. So there are VC files which are generated similar to string here. We are having a different presentation for insertion and deletion nucleotides. For example, this case is replaced, or there is a delete, or there is an insert. So the representation, this is the body of the VCF file. It shows reference allele, the alternate allele. Be replaced by or C is deleted, so you show a previous base and the deletion. If there's an insert, you show first 2 bases and the third base. So this is another example of visualizing, interpreting output data. Since we are lagging behind, I'll just skip these slides. So to summarize, we saw how sequence analysis perform for VCF pipeline, and there are multiple tools that are used in. So we'll be showing you another set of tools. This one I'm showing you using fast cut it up, cut and sample VCF tools. So you have multiple other tools also for the analysis. So what are the applications of VCF analysis? So we can look at disease research and diagnosis, for example, diagnosis of breast cancer or monogenic disorders of cystic fibrosis. Then we can use it for pathogen surveillance, or most widely and most broadly used recently is the SARS-CoV-2 variant analysis pipeline. You can also use it for identifying antibiotic resistance in mycobacterium tuberculosis. You can use it in cancer genomics, where you can look at the somatic mutations or EGFR mutations in lung cancers, and it can also be used in forensics. Next, other popular pipelines in NGS are, so right now we saw this analysis, which is whole genome sequencing, and we look at the variants present. The other is whole exome sequencing, where we target the coding regions of the genome and identify mutations only in protein codes. The next application in NGS is transcriptome analysis, where we look at RNA sequences like mRNAs that are transcribed or expressed in particular physiological conditions like disease conditions and control conditions, and we compare how these are modulated differently between the two conditions. Another application is recently evolving, that is single cell RNA, where again just the transcript of single cell. Another much different that is application is for metagenomic analysis or microbiome analysis, where we look at the microbial diversity in the sample, and we can associate it with the disease phenotype or even with the environment changes that are occurring associated with the system of interest. So these are three major or broad applications of NGS pipelines. So all these analyses can be performed on ICE Cloud, provided you know all the tools that are required for the analysis. So now how is ICE Cloud different from desktop? So firstly, storage. Your desktop is private storage. You cannot share it immediately with anybody. Whereas ICE Cloud provides you secured storage along with secured sharing. Nextly, your databases that you have on your private servers occupy a lot of space, whereas on ICE Cloud, you can request the administrator to actually download the database and keep it in the public folder. Since we saw in the previous talk, Ramji showed you the drives which are mounted on the Docker. So you can see a public drive. In that public drive, whatever databases you want to use for your bioinformatic analysis can be stored. And since they are public, they will not come to your user data. Next, softwares have to be installed from source code or Miniconda on your desktop, whereas in ICE Cloud, you can straight away put Docker image. Access to the software requires customized and support supports pipeline development, and ICE software are isolated into containers, and the cloud also supports zero pipelines. The compute resources are limited and efficient utilization is there on desktop, whereas ICE provides scalable and efficient resource utilization. So to end this talk, to summarize, what are the challenges in NGS data analysis, how ICE will benefit you, what are the basic steps of variant calling pipeline, and we saw other pipelines which can be used or for analysis with ICE Cloud, and what are the differences between desktop. So here I stop my presentation, and I request my colleague Dr. Tina to take over the actual hands-on workshop for VCF analysis. So thank you for your
I request everybody to stay online. We'll have a break of 5 minutes and start again. Okay, so now after some time, we'll be starting with the workshop. We'll be having, this is just a test audio we are trying to check. We are going to have hands-on session on NGS analysis. And to all the participants, we are just checking on our side. We are not starting with the hands-on. We'll meet at 11:30. We'll we'll start with the hands-on by at 11:30. Kindly do not log out. Stay connected to the meeting. We'll start at sharp 11:30. Whatever you hear till then, that's for the testing purpose from our side.
So am I audible to everyone? Am I audible now? So is my screen visible? Okay.
Shared.
Yeah, I have already shared. So good morning, everyone. So by now you all have had some exposure of ICE integrative computing environment that we have developed. And Dr. Sucheta has already covered the next generation sequencing analysis basics. So now we'll start our hands-on session on ICE. And what we will be covering is the reference-based mapping of NGS data using ICE Cloud. And we'll walk through some basics of basics of like NGS analysis, the reference based, and we will be covering like how to fetch data from the sequence read archive. That is the database, and you can retrieve the raw sequences, the raw reads, and the conversion of .sra files, what you will be retrieving to the FastQ files, and then the fast QC of those FastQ files, and then the reference mapping, and the conversion of SAM files to BAM file, and the alignment of short reads to the reference genome sequences. What we have, what I will be explaining, what is the reference genome, and then the variant calling features, and lastly, the annotation. The annotation of those variants, how do we annotate, whether it's a synonymous, non-synonymous mutation, and then the annotation and the visualization using the integrative genomics viewer. Right. So these are the basically the steps what we will be covering. And another is the important aspect is that the data set what we have considered is related to the antimicrobial resistance, and all of you must be aware of this term. What is antimicrobial resistance? That is, the bacteria are becoming resistant to the antibiotics that were previously used to treat them. And there are like various mechanisms of resistance like efflux and the presence of the beta lactamase enzymes and the permeability defect. And one of the major mechanism is the point mutation, the changes in the DNA sequences. So scientists often look at those changes which affect the binding of particular antibiotics. So for our analysis, what we have considered, like our pathogen is the Klebsiella, our data set is the reads, and one is the case, that is the mutant one. Another one is the control, that is the susceptible one. And what we have considered is the colistin antibiotic, that is the last resort of antibiotic. And like if a bacteria is resistant to that antibiotic, that we have nothing to prescribe for the patients. So this is the data set what we have collected from the literature. And we will be investigating two important mutations. Yes. Yeah. So these two important mutations in PMRB genes. This is what we will be investigating using these data sets. And this PMRG is basically the sensor kinase protein. The mechanism of this PMR gene for conferring resistance to colistin is different in different bacteria. But here we will see how these mutations affect the binding of colistin. Right. And in our upcoming demo in molecular dynamics, we will be showing you all the docking, how these mutation affect the docking of that particular antibiotic. So this is all about the data set, what we have collected and we have. And here you can see here in tutorial, like organism is Klebsiella. This is the case. This ID is case, and this ID you can retrieve from the sequence read archive. That is the run ID. And here the control ID. So here you can see the two different IDs are here. One is the experimental ID and the another one is the run ID. And you can fetch your data using any ID, but please follow my steps, what I will be doing in my demo. Right, and this one is the reference strain. This reference strain is the complete chromosome sequence. We haven't considered any plasmid sequence. This is the complete genome sequence of Klebsiella pneumoniae. And these are the variants. And for these variants, these are the literature. Right? We have given the link how these mutation affect the binding of antibiotic. And now the important tools, what we will be considering is the SRA tools, Fast QC, TrimGalore, BWA, SAM tools, BCF tool, SMP EFF, and IGV. These are widely being used tools. This is a like routine practice in clinical settings for the identification of variants. So first, I'm assuming like you all have access to ICE Cloud and you all have your credential, your login credential. So what we will be doing, like in your browser, type ICE Cloud. Oh, sorry. icecloud.in and enter. Here on left hand side, the login is here and type your username and password. We have already provided your username and password. So this is the dashboard, what we have already explained. And this tutorial, you can, like, we have already upload this tutorial in our public drive. And here you can see the ICE workshop folder. Click on this and NGS analysis folder. And here you can see this tutorial. This tutorial we have uploaded and you can use this tutorial later. Right? And now what we will be doing is, so this what I have already explained, like this is the dashboard. And now we will be using first the SRA tools, like how to retrieve data from sequence read archive. And the file you will be retrieving in .sra format, that is the raw sequences. And the common utilities are the prefetch and faster QDump. So faster QDump is nothing but the faster version of FastQ. And then we'll convert the .sra to .fastq files. And now we'll do the analysis. So now go to container and images, right? And now my ICE Cloud is open here. You can first we'll go to home page for your convenient, and then we'll you click on the container and images, right? You all can download the tutorial from here. Yeah, I'll show you once again. Yeah. In storage, then you click on file drive, and here is the public drive, which has been shared with all of you. After clicking, you can see here the ICE workshop folder, and in ICE, NGS analysis folder is there, and you can download your folder, your tutorial here by right click and then download. Right click and download. It will not be open in ICE platform. So are you able to like follow this? Are you able to follow what I'm telling? Like, is there any problem? Anybody facing any problem? Okay, so after downloading, like what I have explained is now we will start using the NGS analysis tools what has already been embedded in this ICE platform. So what we will do, we'll click on this container, and then images. In images, you can see how many tools are there. These are the tools. But for this step, what we will be doing SRA tools to fetch the raw data from sequence read archive. So what I will be doing is SRA and then enter. SRA and then enter. Or you can follow this step like SRA tools. This is not case sensitive. And then launch. Here is the launch button. You can see in my screen, right? This you can see in your tutorial here also. Type your query here that you have to mention SRA tools and then launch container. And you will be able to see this, this type of screen like to create container for SRA tools. And here what you have to mention is the job name. And no special characters are allowed in this text box. So what I will be doing, I'll simply write the SRA tools. Or you can write anything, but this is special characters are not allowed, right? You don't have to change the image name. And here for smooth execution of all the tools, what we will be doing, we will be using 10 GB RAM and 5 CPU. And for every tool, like we have already provided you some quota, we have limited space. And for this, you have to mention like for the 10 GB RAM and 5 CPU for every tool for this demo, right? And having mentioned, you don't have to click on the notepad, your notebook, and then create container. You have to click on create container and this pop-up, okay, on this pop-up. And then the status, you have to check that status. It is still waiting. Now you have to refresh it from here. Now it is running. So please check everyone the status of this screen.
If anyone has
Your
any issue,
status should be running.
if anyone has any issue till this stage, please raise your hand or
The
message.
quota is 10 and 5. So just see if you have redone one more times, then the pod creation might be failed because your limit is 10 and 5. How you can check is on the main page. On the main page, you can see here the instances. Here you see how many are running. You are allowed only 5 and 10.
Right.
If already
So
something
what
is running,
I will do this.
your instances page should be displayed this way. Should we move ahead? Yeah.
Yeah. Okay. So now please check your status and this should be in green color and in running. And then click on that action button, that action, and then connect. I have also mentioned the same steps in this tutorial. Press refresh button for changing the status and then connect.
Now everyone has running status. If any issue, please message.
So now we have connected to the terminal, and here you can do further analysis. Like the first command, what we will be doing is pwd to check in which directory you are in, right? So our first command is pwd Okay. Okay. To open terminal, your status should be running and then click on action. Okay. Then click on the action button, action 3 dots you can see here. Okay. So go to container and then instances. I'm already here.
Yes.
Yeah.
Just for them.
Yeah. And here you can see the status. So now you have created a pod for running SRA tools.
If you are unable to see the status as a running, then the create container, create advanced container image. After image, the refresh button is there. You can refresh it, then the status will update.
You have to refresh your page. You can refresh from here. Your refresh button is just adjacent to this images. This green, blue, and yellow. Okay. I'll repeat steps to create container. Okay. To create container. So first what I will be doing is I'll delete. Yeah. So first I will delete this and I'll create again, right? Yeah. So now I have nothing here, right? And I'll create again the same container for running SRE tools, right?
So hopefully everybody is on this home page at least. So from here, we'll again show you how to create this instance.
Yeah. So go to container and then click on images. Here you can see all tools what we have already on Ice Cloud. And in this search box,
This is only for those who are not able to do it. The ones who have got the status running, kindly don't do these steps. This is for the people who have not created the instance at all.
in search box, type SRA and then run, enter. And you will see this container. From here, you have to launch. After launch, what you have to do, you have to mention the job name and no special characters are allowed, right? So simple, you can type here SRA tools, or you don't need to change that job name, right? If you want, you can change, or there is no need to change, right? And here, what we will be using is 10 GB RAM and 5 CPU for smooth execution of all the tools for this demo, right? You don't have to click on notebook and then create container. Please don't use higher value for RAM and CPU utilization. So we have given you the quota, default quota. So you have to mention 10 GB RAM and 5 CPU for this demo. And then click on create container. Click on this pop-up. And then you will see this screen. Now you have to refresh first. Your status is waiting here, and after refreshing this page, you can see the green color status that is running.
Still not able to launch this. Show them once again where is the refresh button.
So anyone facing any issue?
If anyone is facing any issue in making this instance, then kindly put it in the chat box. Else we'll go to the next step.
Show them once again refresh button. Where is the refresh button?
Okay. Now your status is running, and what we will be doing to connect it to the terminal, here you can see the 3 dots. Click on this and then connect. This right. Yeah. So this is your terminal, and here you can run command related to SRA tools. Right? So now we have opened our terminal, and what we will be doing, please do not run any command, because this command, the upcoming this step will take some time. So please do not run this command. So I'll tell you. Okay. So the first step, how you will download your raw sequences from sequence read archive, is a prefetch. This is the utility of SRA tools. But do not run this command, because this will take around 3 hours. So what we have done, we have copied this to the public drive. Okay. So what you need to have is the case. First one is the case, that it mutated one, right? And second is the reference genome. So what we are doing in our next command is, first you see where you are. Like PWD, you can type here. And this should be like /drive username, whatever we have given you. Now we will be copy this file. First is the reference genome, and this is what I have highlighted it /drive/username. So here you have to mention your username, what we have given, right? What you have created, right? And you copy this. You can copy this from tutorial and then we will paste Control Shift and V. Control Shift and V, and my username is Tina. What we are doing is we have already uploaded .sra file and the reference file in public repository, in public folder, because it will take some time to prefetch it from directly from the sequence read archive. So I am giving you the path from where you can retrieve these files and copy into your folder. So this is what we are doing. We are just simply copying it from one public drive to the your path, right? Private. Yeah. Now enter. And here you can see whether it is copied or not using the ls. So your first file, that is the reference genome file, is here in your public, your private folder, right? Again, go to tutorial, and what we will be doing, we have to copy .sra file, that is our case. Again, we are doing the same thing. We are copying from the tutorial and Control Shift and V to paste. Control Shift and V. And then enter. So this will take some time. This file is like huge size, so this will take some time, right? So everybody is following these steps, or any issue, anyone facing any issue? If you are facing any issue, you can paste it on chat.
While copying, the destination folder is /drive/ the username that you had created. Like in this case, the username is Tina. So it is /drive/Tina. And if at all there is any confusion with the username, once you are in this terminal, just type pwd and enter. The path you will see, that is the path that you have to paste here.
So this CP, then space is there, and then path to our public drive where we have uploaded this .sra file, and then the path you are getting from pwd command.
This command will give you the path. This path is to be pasted over here.
Control plus plus.
हाँ लेकिन फिर मुझे प्रॉब्लम हो जाएगा ना.
If you have this, you can just check the PWD. It will show in which drive you are still. Then you need to be in copy in your the present working directory drive. This is the file you need to copy. If you are unable to copy, then check once again, ls I have copied the 2 files that is present in my directory. If you are unable to copy, then just instead of giving your destination /drive/ username, you can just give dot. So dot means it will come into your current directory only, and then you can enter. So it will copy the existing file. So it is around 1 GB. So it will take some time. So give the time, then you can check what the files you have downloaded. I think everyone has downloaded these 2 files. If not downloaded, please message. So I think everyone has downloaded because no one have not got any message. So the next step we will
You can see your file using ls command.
Yes.
ls command can be used to see whether you have copied your file from public drive to your current working directory.
Now go to another list.
Right? So now we have the case file and the reference genome file, right? Now go to tutorial, and what we will be doing is we have to convert this .sra file to FASTQ format, right, for further processing. So what we will be doing, we just simply copy this, what has been highlighted in orange color. So you just copy this command and Control Shift and V in your terminal. So you already have this SRA file in your folder. What we are doing is FasterQ dump file, it will split because it is a paired end sequencing. So it will split in forward reads and the reverse. Fine. So this is the command and these are the threads. So we have already mentioned 5 CPU we are using. So we have mentioned the threads we are using 5 and FasterQ dump. It is nothing but the faster version of FastQ. And then enter. So here is the command, right? Are you able to run this command or anybody facing any issue? So this FasterQ dump is the common utility of SRA tools. So we are using the same package SRA tools, and here we have used prefetch. Prefetch we haven't run because it will take some time. We have copied our file from public repository to current folder. And now what I'm doing is FasterQ dump utility I'm using from SRA tools. You can type your issue in chat box also, so that we can resolve. If anybody facing any issue,
In chat, the copy command also and this command also.
okay.
Right. I'm telling you.
Okay. So you don't have to mention the reference genome file. Here we are using .sra file. In your current folders, you have two files. One is the reference genome .fa and second is the .sra file. So you don't have to mention that .fa file. That is your reference genome. Look at the chat box. One second.
Complete till dot. Select it also.
क्या करना है?
हाँ. After space dot is also there. So complete till dot.
Okay. So in this command, like copy, and then we have given the path where we have already uploaded that reference genome, and then space and dot. So you also have to mention that dot, that is your current directory. While working with Linux, that dot represents the current working directory. So you don't have to mention, if you are not able to do pwd command, or you are not able to mention that /drive or /username, or you don't know the username, you can just simply mention this dot. cp space, then the path we have given, space, and then dot. This dot is your current working directory. And then enter. Right?
If anyone has just raise the hand.
If anybody facing any issue, please raise the hand. So, Dr. Anisha, please mention your issue like in chat, you can type. No such file. No such file.
So please copy properly. Everyone has copied source only. Type publicly.
Okay, I'm repeating these steps again. So where is this file? Like here is, I have already. So here, click on the storage, file drive, and this is the path. When you click, if you are not able to see the file, you just click on this public drive. And then here we have created one folder that is ICE Workshop. Inside that, you can see one more folder that is NGS Analysis. Click on that. And here you will be able to see that file. These two files only, .fa file, that is your reference genome, and .sra file that we have retrieved from sequence read archive. This one.
Yeah.
Please check your ID because there are two SRA files. Here two SRA files, but you have to copy only one SRA files, and that is ERR 120779995, because that is your case, what we are analyzing, right?
Hello, Dr. Anisha. Yeah, are you on the terminal that is currently being displayed? This is the exact instance that was launched. If you are on the terminal, can you just reply yes or no?
Oh, yeah, right.
So this command is taking some time. Command will give you two files. First one is our ID underscore one dot fastq, and second is the second file, like ID underscore two dot fastq. So we are working with the Illumina paired end sequencing. So what we will be getting, this is splitting the sequencing file in forward and reverse reads. So after getting FASTQ files, it is taking some time. So let me just explain you the next step, what we are going to do. After getting these files, what we will be doing, the FastQC will perform the quality control checks on raw FastQ files, what we have generated using .sra files. So our next step will be FastQC, quality control. So the common utility is the fast QC that I have highlighted in red color. And this is the overall statistics, what we will be getting. So the file will be generated in HTML format, and you can see the basic statistics of your raw reads, like per base sequence quality, per sequence quality scores. You can see per base GC content, per sequence GC content, per base N content, adapter content, and the overrepresented sequences. So in next step, what you have to do, we need to remove the low quality reads, or if any adapter is present, so we have to remove that. And for this, we use fast QC. And since this is a separate tool, so what you have to do, not right now, but I'm just telling you, in next step, what you have to do, you have to delete your previous pod, what you have created for SRA tools, and then we'll create the new one for the fast QC, because this is a separate container. Okay. So here, files are here. Now, the command is executed. Complete. Yes. Now you can see these two files, .fastq, id underscore 1.fastq, and id that is your run ID from sequence read archive, underscore 2.fastq. So these two files will be generated using fasterq dump. Now we will be working on these two files. You can see here the tutorial and the fast QC, what I have already explained. And if you are done with these steps, what you will do, you have to delete your previous pod. So what we will be doing next, so this is what I created for running SRA tools. Here you can see the image name. So I'm hoping you have only one pod. So what you will do, go to action button, action, and then delete. Your files will not be deleted in your folder, right? We are just deleting the pod, what we have created for SRA tools. Okay. So here all files are here. So you go to home page and storage and then file drive. And here you can see your files. Your files are generated. Now you have 4 files. One is reference, second one is .sra, and we have converted .sra files to fastq files. And we have generated these two because we are working with the paired end sequencing. Right? Now in next step, we will be using these two files and we'll check the quality of these reads, whether we can go ahead with these reads or we need to trim, right? And what we will be doing, now go to container. Here you can see all your files are here, right? Now you can go to container and then instances. This is my previous pod, what I created for running SRA tools. And now, because we have limited quota, so what we are doing is, we are deleting this. Go to action button and then delete, right? And then yes, delete it. In pop-up, click on yes, delete it. Okay. Now the pod we have created for SRA tools has been deleted, but your files are still here. Your file is in your current folder, right? And you can refresh from here. After deleting, it is showing the status terminating. You can refresh it here. Now my pod is removed. And since I will be working with fast QC, so what I need to do, I need to create a pod for fast QC tool, because it is a separate tool. It is not a part of SRA tools, right? So what I will be doing, go to container and then images. Fast QC.
टाइम स्टार्ट है तो डायरेक्टली गो टू प्रिंटर नाम से पूछूं हाँ आप करो मैं ओके बोलो
Okay, fine. So our next command is fast QC. I have already explained you the statistics and the HTML file we will be getting using this command. So due to time constraint, what we are doing is we run this command. So we have already deleted that previous pod, and the fast QC for this demo we are not running. What we will be doing is direct trim galore we will be using, right? So what it does, this will remove the low quality base and the adapter sequences, right? So what we will be typing in text box is the trim galore, this one. So now go to container. So you will be running steps from here. Go to container, instances, action, delete. Right? From here you can see your home page and then container and then images. So here you can type trim and then enter. Due to time constraint, we are not running command related to fast QC. And I have already explained you the basic statistics, what we will be having. What we are doing is we are skipping this step and we are directly running the trim galore on these files, what we have generated from our previous command, right? So what you have to do, you have to first delete your previous pod. I'm assuming you have already deleted your previous pod. And then you have to create your new one for trim galore, because this is a separate tool. We are not working with the SRA tools, right? So what you have to do, click on container and then images, and in search box, you can type trim and then enter. And then launch. And the same steps you have to follow what you have followed for SRA tools, right? And this you can use default. And image name, you don't have to change this. What we have to change is the memory allocation for image, that is 10 GB we are using. And the CPU utilization is 5. Right? At this page, you don't have to make any changes. Only mention 10 GB RAM and 5 CPU we are using for running the trim galore. And then create container. You have to click on this green button for creating the container. At this pop-up, click OK. And now you have successfully created, but your status is still waiting. Your status is waiting, so you have to refresh from here. Again, you have to refresh from here, and after refreshing this, the status is now green, and now you have successfully created a pod for fast your trim galore, sorry, right? And now click on this action button and then connect, right? Again, you are in your working directory, right? Type simple ls. Now everyone is with me. So type ls command, and here you can see your previous files, what you have generated, right? So you have only deleted that pod, but the files are still here. Okay. So what we will be doing, so we have to run this command for trim galore. Now you have this file 1.fastq and the 2.fastq, and for trim galore we need both files. Okay. So what you have to do, just simply copy this from tutorial, and what you will be doing is Control Shift and V. Control Shift and V to paste this command on your terminal, and then enter. Chat box. I'm also pasting the same command in chat box. Right?
It will take around 10 minutes. Till that, you can explore the ice.
So this command will take some time. So here in this command, what we are doing is we have mentioned paired because we are working with the paired end reads. So these are your forward reads, first file, and second one is your reverse reads. And fast QC, we have mentioned which tells the trim galore to run fast QC automatically after trimming. And for trimming the Fred score, it is, we have just using the default values, that is above 20. So you will be getting .fq file after running this command, and the reads have already been trimmed. And we have used default Fred score, that is greater than 20. It is still running. If anybody facing any issue, you can type in chat box or raise your hand if you are not able to follow these steps, or if you are not able to follow, like you are not able to generate these files. So whatever files we have created so far, so far we have uploaded all of these files in public drive. If you are not able to run these commands, so you can get these files from public repository, public folder, public drive, right? So if you don't have those, so I'm sharing my screen. Here is storage and then file drive, and here public drive is here. Click on this public drive. ICE workshop. Click on ICE workshop. And then NGS analysis. Where are the files? Yes. Where have you copied these files?
पूरे सारे और 5 फाइल इसका कॉपी कर रहा हूँ बाद में डाउनलोड करेंगे.
Okay. It will take some time. I'll let you know. I'll let you know when these files will be copied in this public drive. So it is still running. So what we have done so far, we have retrieved data from sequence read archive in .sra format. Then what we have done is conversion of .sra to FASTQ, because we are working with Illumina paired end sequences. So we are getting 2 files. We have skipped one step, that is FastQC, because it will take some time. And directly we have run the trim galore for trimming the adapter sequences and the low base quality sequences, right? And still it is running. And after running this, you have to follow same steps. Like now we will be working with BWA tools. So what you have to do, you have to delete your previous pod, what you have created for trim galore. And then we will be creating one pod for BWA. And what we will be doing, we will prepare our reference. As Sucheta has mentioned, we have to prepare our reference genome first. We will be doing the indexing. We will be converting the genome sequences into efficient data structure so that it can process the short reads for further alignment. So what we have to do, we have to prepare our reference genome first before alignment. So this we will be doing using the BWA tools. So if you are not able to generate these files, we are copying these files, .fastq files in public directory, in public drive. So you can get it from public drive, right? And all of these commands are mentioned in tutorial, and you can perform these steps later also. Or if anybody has any issue or any question, then raise your hand, or you can type your query in chat box. So this command is taking some time. So we should wait for another 4 or 5 minutes. So in this command, we have used the default value of Fred. And this command will remove the low base quality and the adapter sequences. So after running all of these commands, you should remove your intermediate files like which are not required for smooth execution of these tools. We will be doing this later, but I'm just telling you. So by the end of this tutorial, you will get to know how to execute end-to-end NGS analysis using this cloud platform, right? So now after trimming, it is writing validated paired and read one to that file, and reverse one is in second file. So what we are doing is we are processing raw reads to investigate the mutations which have already been reported and conferring resistance to colistin, right? So for processing raw reads to annotated VCF file, we are doing these steps. And so far we have used SRA tools, Fast QC, Trim Galore. These tools we have used, and now the next step is the preparation of the reference genome. A reference genome is your complete chromosome. We have not considered plasmid sequences or any other sequences. This is your complete genome sequences of Klebsiella. The command is still running. We should wait for 1-2 minutes. Then we'll go ahead for BWA for reference preparation. Neeraj sir.
Yeah, expected output something.
Neeraj. One second.
Yes, we have actually increased the quota for you. Now you can run up to 10 CPUs and 40 GB RAM. So kindly let us know if that is not the case with you. You can see that in this over here in my quota here you can see. So currently this is for our admin usage, but you can see 10 CPU here and here you would see 40. Just check this, and if this is not the case, click on request quota here and send it to us. And this particular usage of 10 CPU and 40 GB is limited to today's workshop. After the workshop is complete today, it would be revoked back to the original of 5 CPU and 10 GB. Hope everybody is clear. In order to check your quota, go to this tab, go to my quota, and here you can see it. Okay, here in my quota, you have to go and check for the quota. That is for the request, but this is what here you should get 10 and 40 GB. Over here, my account, and then quota. Here you can see. If here is not the case, then you have to request the quota by going in this particular tab.
It's still running.
It is still running.
20% gone. So take some time.
So you can see that even though if you have allotted 5 CPUs, so for 2 GB files, it taking you a lot of time. So for that, you require a bigger HPC and more number of nodes. Generally, for normal, whatever practical applications you are running, so the file size would be much bigger. And if you have a less number of cores, that won't be working. It will be taking huge amount of time. So you require bigger and bigger HPC machines for doing this analysis. And the memory also required is more for genomics data. So this is just to show you that why you require an HPC to perform such tasks.
45% US.
Yeah. So it is about to complete like 45%, then 50%. So we should wait for 4-5 minutes. Then we will be able to get our files for further processing.
I hope everybody is able to see that their results are getting completed 80% or 70%, something of that sort. Huh?
इनको बोलो स्नैपशॉट ले लो स्क्रीनशॉट पेस्ट करो चैट में
अभी इसको कितना टाइम लगेगा?
It is about to complete.
So you can see that at our end, it's a 100% analysis is completed. Now another next step is going on. So that will take another 2 minutes. So as this is a demo and we have allotted only 5 cores, if we would have allotted more number of cores, your job would have been completed faster. These things. Solution कुछ है क्या?
algebra. Last.
उसको कितना टाइम लगेगा? वर्क फ्लो का बोलो कि ये अलग अलग स्टेप्स कर देना तो इसका एक पूरा कंप्लीट वर्क फ्लो भी बना सकते हो इसमें एक के बाद एक स्टेप बोल दिया.
So since this workshop is to make you familiar with what is ICE and how to handle data using this platform, so we are what we are doing is we are running individual tools, right? Creating pod, then again deleting pod and this. So what you can do is we have commands already here, and what we can automate these steps, right? We can make a bash script and you can run this script directly in a single run. You don't have to go here and there for deleting and creating pod and this. So here we go, like this analysis is completed and we are now able to create those files which are required for further processing, right? And these 2 files are here. This one for the forward and this one is for reverse reads. So these 2 files are now required for further analysis. And 1 second, my colleague is about to.
Now, as in earlier step, we did not do the fast QC, but at the running of the trim gallery, we have used -fast QC parameter. So you can see here the .html file has been created. So later on, you can check what is the quality, and when you will get the time, the fast QC commands everything present in the tutorial. So you can run yourself and you can check what the earlier quality was there and how the giving the quality value the cutoff value the 20 has changed your reads, and also the number of the reads and the quality of reads. You can check it. Now we will go the next step.
So our next step is the preparation of the reference genome, and we will be using BWA tool, that is another tool. And the first step is what you have to do. You have to delete your previous pod, what you have created for this trim gallery. So what we will be doing, home page, and then click on container and then instances. So here I created that. What I need to do, click on these 3 dots, action button, and then delete. Right? The status is terminating. Now you have to refresh it from here. Now I have deleted the pod created in my previous step. Now we will be using the BWA tool. So what I need to do is we have to create a pod for BWA tools. Go to home page, container, and then images. Type here BWA and then enter. After entering, like, you have to launch this. And to create container, this can be default, this can be default. And what you have to change is, like, we are using 10 GB RAM and 5 CPU. The same step we did for previous analysis, right? And having completion of this, what you have to do is to create container for BWA. So
Don't change image name. We got someone has error due to they have changed the image name. If you will change the image name, then the Docker cannot be pulled because the name of the Docker you will change, then it cannot be pulled from the source. So please don't change the Docker image name.
you only have to mention that you are using 10 GB RAM and 5 CPU.
Okay.
Right? Status is waiting. Now you have to refresh this. It is running. Now I can connect this to BWA terminal. Right? And here it is. So if you run ls,
So PWD, everyone PWD seconds. Requested to do everyone PWD. Now you are in /data. So now you have to go into your home directory. So to go home directory, please type cd slash drive slash, and then you can use tab. Or if you know your name, then it's okay. In the Tina and please tab. Tab.
PC और किसी का भी हो सकता है.
So when after that you will press the tab button in your keyboard, then whatever the directories are present inside the drive, you will able to see it. So public, shared, and whatever your name is there, it's present. So now as my name is Tina, so I have to type T. Now once again I will use the tab key button. So I will go into the Tina. Now I will enter it. Now I am in my home directory. Please use PWD to cross check in which directory you are.
Type PWD.
Now you can see also the PWD. I am in /drive/Tina, and the files created in the previous commands are present here. The commands used, we have the trim galore, and here we have the our sample name _1_val_1.fastq and fastq2 file, both the parent we have. So now we will go to the third step.
So for this, what we'll be doing is to use this command. BWA index and then your reference. Now we are preparing your reference sequence, right? For alignment. So we'll copy this and Control Shift and V. And then enter.
The reference file will be created. So why reference is needed? Because for the alignment purpose, we need to lookup table for the reference, because this is the small bacteria, so a small genome is there, but at the time we have genome file in GBs.
So need to index the file for the faster lookup for the alignment purpose. Now index is completed. Now we will go to the alignment process.
Now you can see your files using this command and this .bwt, .bac, .annotation. This I have already mentioned in this. So these index files are not human readable and they are binary or structured data files used internally by BWA for fast read alignment. So you don't have to check what is generated. Okay. So now our reference genome we have prepared. The next step is running this command, aligning paired end reads using BWA-MEM. BWA-MEM is the utility and the command part I have written here. But first I will execute this command, right? And you will copy this command. So since we are using the same BWA tool, so you don't have to delete your pod or create your pod for some for another tools. We are working in the same pod, but you have to run this command. Simple copy this command and Control Shift and V on your terminal and then run. Are you able to run this command? So in this command, BWA-MEM is the utility of BWA and that is suitable for Illumina short reads. -T5 we have used for 5 CPU threads we are using here. -R add a read group header to SAM files. Print more verbose output like the intermediate commands like you can see on your terminal. And then output file is .sam file. It will generate .sam files. What you have to do is to convert SAM to BAM in coming step. This is your reference and these are the forward read file after trimming and this is the reverse what you did using the trim galore. So this is what we have executed, right? And this slash is only representing the new line. It doesn't make any difference like if you
Escape character for the new line.
This is the escape character for the new line, right? So you only have to copy this complete command and you have to run on the same terminal where we have generated our indexing for reference file. And here in this command, if you can see here, this what I have highlighted here. When we use multiple sample, you have to mention this and the meaning is mentioned here. Like ID, you have to mention read group ID, sample name, platform, what platform is there, library name, platform unit. So this is all you have to give if you are working with multiple samples. So I've just mentioned to make you understand like how we can run this BWA-MEM. But we are working with a single sample. So if you will not be providing this particular tag, so that will be fine. But we have given the entire information so that you can create your automated scripts in future, right? So what you will be getting is SAM file. So after this step, we have like .sam file, and the next step is convert, sort, and index the alignment file. For this, you have to use SAM tools, and this is the another package. So what you have to do, you have to delete your previous pod, and then we'll be creating the new one for SAM tools. So this is about to complete. Like it will take some time. .sam file, right? So simple. What we will be doing is to create this, and these commands won't take much time. These 3 commands you have to run on SAM tools pod. Yes. This won't take much time, and after that we will be generating the VCF file. ये will not take.
एक ही पहले पेश करके डॉट डाइसेक्ट करके कैसे बनाता है वो भी एक रास्ता है।
बैश बोल रहे हो
अरे उसमें टाइम मिलता है।
Okay, so we have generated those files like .sam files. One second. So here I have generated this .sam file. What I have to do now, create container for SAM tools. But what you have to do, you have to delete your previous pod, go to container, instances, and then we'll delete this one. Action and delete. The status is terminating. You have to refresh. Now go to container, images, and SAM tools. Now we'll be working with SAM tools. So what you have to type is SAM tools. Container, images, and then SAM tool in text box. And here you have to launch. The same step, we have already done these steps in our previous commands. In our previous steps, sorry. So this is default. This image name is default. What you have to do is we are using 10 GB RAM and 5 CPU. And then we are creating container for SAM tools. Click on OK. Your status is waiting. Now you have to refresh. After refreshing, click on action button and then connect. Now you have created a pod for running SAM tools. Click on LS. So you are in a correct directory. You have all of your files you have generated, and then go to tutorial. And what we will be doing, we'll copy this command. So this command is converting your SAM file to a BAM file. Just copy this command. I delete this. And Control Shift and V, right? And you already have .sam file in your folder, and you have to run this. I have also pasted the same in chat box, so you can copy this command.
This will convert SAM file into the BAM file. SAM file is a huge file. It's around 11 GB file. It will take our disk spaces, and all the next tools are using the BAM file, sorted BAM file. They does not directly use the SAM file. So we need to convert BAM file into the SAM file into the BAM file.
So it's still running. Next one is this command, sorting the BAM file. And this is essential step before indexing, variant calling, or visualization. So we have to sort that BAM file using this command. The second command is also a part of SAM tools. So we don't have to delete the previous pod and create the another one. We will be working in SAM tools, what we have already created. So now we have completed this. So here we have the .bam file. In our next step, what you have to do is to sort the BAM file using this command. You just copy this command and paste it on the same terminal. Copy here and paste this command in same terminal, right? So the input is .bam file you have generated in your previous step, and it will convert it into the sorted BAM file. And then enter. After sorting this, what you have to do, we will be using SAM tools index. Like it creates an index file .bai for your sorted BAM file. And this BAI file is required for further analysis. So we have to perform this step for further processing, right? So I'll copy this command from here. So this command will take around 1-2 minutes. So you have to wait for some time.
सब लोग एक साथ रहेंगे टाइम लगेगा
हाँ,
उसके बाद
otherwise बस इसके बाद हमें वेरिएंट कॉलिंग
कितना टाइम लगेगा
वेरिएंट कॉलिंग में लगना तो चाहिए क्योंकि हमारा yeah. So next what we are doing is just copy this command and paste it on the same terminal. Control Shift and V and enter. The command is executed and you can see your files here. Okay, so I'm also pasting this command in chat box. And our next step is to use BCF tool for variant calling, right? So what you have to do, because this is a different container, so what you have to do, you have to delete your previous container, what you have created for SAM tools. So go to home page, container, instances, and then action and delete. Status is terminating and you have to refresh this. Now I have deleted the previous pod and I have to create one container for BCF tools. So go to container, images, and then BCF. You have to launch this here. Job name is default. Image name is, you don't have to make changes. What you have to mention is we are using 10 GB RAM and 5 CPU. And then create container. You have to refresh this. And now here you can connect your terminal. Right? And here I have all the files. So what I have to do is to copy this command. We already have this sorted.bam file we have generated in previous step, and it will generate .bcf file. Again, this is a binary file. You don't have to check. This BCF file will then be converted into the BCF file. Control Shift and V.
Right click, paste.
Control Shift and B. Control Shift, Control Shift and V, sorry. And then enter. So in our next step, we will be converting this binary call format file to .bcf file. And we will be able to generate VCF file of our raw reads using the reference genome. So it will take some time.
हार्डली 10 मिनट्स ये बोलो कि बड़ी एचपीसी होती है तो आपका ये पूरा टास्क 10 मिनट में होता है, बाकी टाइम 1-1.5 घंटा तो लगा है टाइम में बता।
So it is taking time because we have given you the limited quota. You have limited CPU. So you need like HPC system for running this command faster.
This entire job would have been completed in 10 minutes if you have bigger machines. So here it took almost around more than 1 hour. But on bigger machines, the entire workflow can be completed within the 10 minutes. And this was a small file. So we are talking about the 10 minutes. Generally, you have a bigger files, so they take more amount of time. And so for that, you require bigger and bigger HPC machines. So there comes the role of this NSM and this ICE. So there you can have a larger quota and larger number of CPUs to run your jobs.
And here we have all the customized Docker images. So you need not to have installation process worries and all that things. So on just one click, the entire tool and everything with the environment is loaded. You have to just type your command along with your input files. You get all the output files. And one of the biggest beauty of this ICE is that in the next step, you will be seeing that when you are using any HPC cluster, you don't have a just a visualization facility. But here in the ICE, what we provide all at once, one-stop solution. It is one-stop solution. So here you can run all your tools also. You can do analysis also. In the next session, we will be seeing how using the Jupyter notebooks, you can do the analysis of your data if you have your own data. And you can do the visualization also. May it be a genomic sequence, maybe it be a structure file. So you can do all the analysis and all the visualization at one on one this cloud, ICE cloud.
So now we have to generate our VCF file from .bcf file. So copy this command and Control Shift and V in the same terminal. Right? And this command, this file is here. Now we have generated .bcf file. So you can see here, one second. What?
Yes.
Okay.
Yes, mute.
Okay. So what we have done, we have created our own database for reference genome using this command. And the next step, we are using .pcf file and we are annotating that PCF file using the reference genome. So since we have already given the accession number, NCBI accession number of the reference genome. So what it is doing, it is fetching all the data required for the annotation of BCF file. So in next step, what we are doing is you copy this command and paste on the terminal. Or this is the same tool. So what I'm going to do is to run this command, right? So it will generate VCF, annotated VCF file. Right? Now annotated VCF file is generated and here you can see. And what we will be doing, since we have in my starting, I mentioned that we will be considering PMRB gene. So I know the exact location where this PMRB gene is present in reference genome. So this is just the coordinate of that gene. So what I'm doing is I just want to see whether the mutations are present or not. So what I'm doing is just copy and Control Shift and V in same terminal. So here it is. Oh, 1 second. Yeah. Okay, 1 second. And then grab. This is our coordinate, right? 1738545. Now enter. So this is your output and this is the annotated VCF, and here you can see your mutations. Whatever we have mentioned in our tutorial that we are investigating these mutation in our file. So from raw sequences, you are able to get the annotated VCF file based on the reference genome, and these are your mutations. So if you see here, this is the first mutation, and for second, the coordinates are 1738575. So here I will be mentioning 38575.
Neeraj.
So this is your second mutation you can see here. The arginine 256 glycine. So from raw sequences, we are able to annotate and identify the mutation in the raw sequences. And this case is resistant to colistin. So these two mutations are responsible, which affect the binding of colistin to that PMRB genes. So in our next step, what I will be doing, I'll use the IGV, Integrative Genomic Genome Browser, I will be using for the visualization of these annotated VCF. Okay. So now what I have to do, I have to again delete that pod. अच्छा
हाँ उनको मिल गया ना बोलो कि IGB से visualize, you can visualize it.
Okay. Okay. So due to time constraint, we will not be doing this IGV step. This is the last step, and this is very simple step. And I have mentioned Yeah, everything, each step I have mentioned in tutorial, so you can see. But like now this tutorial was to make you all understand like how to execute end-to-end analysis of NGS using this platform. So this is all about this tutorial. Now our next tutorial on molecular dynamics will be starting after lunch.
One more thing, your logins would be active for 1 week. So you have all the tutorials at your hand and you can download and you can run the same steps. And if you have any issues, you can mail us. We will reply you on that. Regarding if you have a domain related issue, running anything on the ICE cloud, any issue if you have, just let us know. And the last step, what visualization has not been shown, you can try on your own. If you have any difficulties on that also, you can mail us. Now we will be having a lunch break. We will be meeting around 2:30. Yes, 2:15. We will be meeting around 2:15 back, and then we will be moving from sequence to structure. There also we will try to show you how to visualize the structure, how to run the simulations, and if time permits, we will show docking also. So you can join post lunch at around 2:15.